Well, I'm going to round out the Illinois hour, I guess, and I have the challenge of getting us back on track, getting us to lunch here. So it's an honor to have that to look forward to. I just wanted to thank the committee and for this opportunity to speak today. I talked about this last year, but I have the unique opportunity of having attended this particular meeting for the last 14 years since its inception. So it was Dr. Adiola who brought me here as an undergraduate student at that time. So for all of you students who are in the room, we want to see you up here sometime. We want to see you making these, getting into this field, continuing on this in this way. I missed the millennial generation by about a month, but I am a techie, and am one of those young faculty who are watching as our students transition in the way that they communicate in that way. And it is certainly a challenge in the classroom to be able to keep up with that and to be able to effectively communicate in so few words. So what I want to talk about today is a story that we've been looking at for a number of years. And this is one that is based on Dr. Baker's favorite word in serendipity. So this is a project that was I was brought in by Dr. Pettigrew. And it really is going to get at the added value of soybean meal. So building upon what Dr. Stein just talked about in the nutrient composition, what are the other benefits that we may have of including soybean meal in the diet of a young pig, specifically when we've actually activated the immune system? So up until April of 2013, PRRS was one of the major diseases that we were talking about. Now, I don't think we have any hope of using nutrition to actually augment PED, but in this way, we know a lot about the PRRS virus. So it's been studied for many, many years in terms of what are its effects from a veterinary perspective, but here we're looking at the immune activation specifically. So very highly prevalent disease in the swine industry. And from the growth perspective, this is causing respiratory distress. We're getting activation of macrophages within the lungs, and in that way ultimately setting off a cascade of cytokines which is going to reduce the performance of pigs. So it's been estimated that PRRS costs the industry about $660 million a year in unrealized performance. So pigs getting sick at a low or high level in this way, so production losses in the $660 million range. We think there's a possibility, or we hypothesize there is a possibility for specific components in soybeans to be able to affect this immune response. And if you look at the literature, especially on the human side, we can see that soy has been studied for a very long time in terms of its isoflavone content. So those isoflavones, including genistein, which I'm showing here, have been studied quite extensively, mostly in vitro. So in a cell culture model, we can show that these isoflavones can affect how a virus binds, its ability to infect the host, to even promote an immune response in the host. So in this way, there's a very good review article on the effect of isoflavones in a viral infection in that way. But if you look at this paper, this review, there's only one in vivo study that they cite where where isoflavones have been tested, and that was in the pig. And that happened to be in the work of PhD work from Dr. Laura Greiner when she was working at Iowa State with Tim Staley. So I pulled out one graph from from her work where we're looking on the left axis at average daily gain of these pigs. What we've done, she's done a dose titration here from 0 to 800 milligrams per kilogram of pure genistein. So genistein being that isoflavone that's found at one of the higher concentrations in the soybean, but this is purified genistein that was purchased from a chemical company. So in this way, looking at these blue bars, we can see that these pigs which were infected with PRRS in an acute model, when they received genistein, we had some increase in average daily gain when providing between 200 and 400 milligrams per kilogram diet. Now on the right axis, we're looking at the concentration of virus that was circulating in these pigs. And in this way, you can see in the black line, we're getting a dose-dependent response, again, at 200 and 400 milligrams per kilogram body weight. So this was some of the earliest evidence to suggest that isoflavones, when fed to an animal in an acute infection, can alter the viral response and ultimately have an effect on performance of those animals. Now, this work wasn't really followed up on for many years. That was 2001. Dr. Greiner had gone through both genistein and daidzein, looking at them independently. Where this came back around, and that serendipity came in, was when Dr. Dean Boyd was looking at, on a commercial scale, investigating lysine requirements in grow-finished pigs. So they were trying to optimize in this way whole body accretion, or whole body growth versus carcass growth, and identifying the SID lysine requirement from those two measures. What happened was, on the commercial scale, 2 of the 3 barns of pigs broke with PRRS virus and also circovirus at the same time. Now, Dr. Boyd didn't give up. He analyzed those data anyway, didn't just throw them away. And what he noticed was that there was a difference depending on the composition of the diet, or where the protein was coming from in that case. So what I'm showing in this graph, we're looking at average daily gain on the y-axis here. And what's in the red bar are diets in which L-lysine was supplemented to produce the gradations in total dietary lysine. So we're going from 0.65, this was true ileal digestible lysine, all the way up to 0.95, so 4 gradations. And the question is whether this lysine comes from L-lysine or if we just increase the concentration of soy in the diet. So importantly, at each one of these, we have equal contributions of ileal digestible lysine, but that's either coming from a crystalline L-lysine or from soybean meal. These diets contain between 13%, what we call the low soybean meal, up to 20%, so those are the red bars. And in those where we increase the amount, where the investigators were looking at the increase in lysine due to soybean meal, we went from about 19% all the way up to 32% of the diet. What you'll notice is that the blue bar is higher than the red bar in each case. What that's suggesting is that even when we have equal concentrations of lysine, in pigs that have been infected with PRRS and a more complex model than just PRRS, soybean meal is increasing average daily gain of those pigs. So that then built upon what we had seen previously from Dr. Greiner's work, in that perhaps there was another component within soybean that could be affecting this immune response. So Dr. Boyd also presented some of this at Midwest meeting this year as a follow-up to that original work back in 2012. I'm suggesting again, on the commercial scale, that dietary soybean meal appears to have beneficial effects when pigs are infected with PRRS. Now, Dr. Pettigrew had done some of the initial work on this and presented this in abstract form back in 2012. Dr. Roca at the time was working on this at Illinois, and in this case, what we call the high soybean meal diet, when fed to pigs in that acute infection model, increased feed efficiency from 642 to 746 grams per kilogram during that first week post-inoculation. So that brings another component into this, is the timing of when that response is actually happening. So this earlier work suggested that that was happening right after the pigs were inoculated with PRRS. In addition, they noticed that on the clinical side, rectal temperatures were actually lower in pigs fed the high soybean meal diet at 3, 5, and 7 days post-inoculation. So here they were using rectal temperature as an overall measure of clinical sickness in that way. And so by having a lower fever, essentially, that it's suggesting that soybean meal could again attenuate somewhat that immune activation. So the objective of the study that I'm going to talk about here is really looking at just that, looking at the concentration of soybean meal in the diet, basically on additional immune parameters than we had looked at previously, and also the growth performance of those PRRS-infected pigs. So, an acute infection, we hypothesized that if soy had added benefit, it would either support a more efficacious immune response, and if it were able to do that, perhaps rescue some of the growth performance that was lost due to this immune activation. So this was a pretty straightforward study. We used 64 weanling pigs from the University of Illinois herd. They were split sex, so 32 barrows and 32 gilts. At weaning at 21 days of age, we brought them into our disease containment chambers. So bring them into campus in a place that we can control the environment more closely in terms of this infectious disease. They were acclimated to that facility for 1 week, so placed on a very common starter diet for these weanling pigs, and then allotted based on litter, sex, and body weight during that time. So we really had a factorial arrangement here. We either had low soy or high soy, where we increased that from 17.5% up to 29%. And then those pigs were either sham inoculated, which means they received a PBS solution in their nose, or that PBS that had a field isolate of PRRS in it. So we have our control animals versus those which are infected. And just to orient you to how we actually run these types of studies, we have disease containment chambers that are each independently HEPA filtered. So we have two hallways, which shown here. The blue hallway is our control. So all animals in this hallway are going to be our control animals, and those are separated from pigs which have been infected. Now that's necessary just because we can't have all of them in the same room. We can't have crossover of this disease. But each of these hallways has 8 individual chambers, and within each chamber, we divide this into 4 individual pens. So in this way, within each chamber, if all of these are control pigs, 2 of these pigs, 1 barrow, 1 gilt, received the low soy diet, and the other 2 pigs, 1 barrow, 1 gilt, received the high soy diet. So again, this breaks down into 4 categories, and we had 16 pigs in each of these 4 experimental treatments. So control pigs fed low soy, control high, infected low, and infected high soy. Our timeline here started at weaning, so at 21 days of age. We brought them in. Here's their acclimation period. So this is from -14 to -7 days post-inoculation. So importantly, we started our dietary treatments 1 week before we caused the infection. So they started receiving either the low or the high soy diet here, where the green bar is, and continued that for the next 21 days. Here is 0 days post-inoculation, so at the time when we actually infected those pigs. So we have a 14-day period where we are capturing information from them, and we're focusing on day 3, day 7, and day 14 post-inoculation in that way. So again, these pigs were intranasally inoculated with this PRRS. If we look at the diets, these were formulated based on commercial standards. Basically, this was a starter diet for that second phase, where all we're doing is altering the concentration of soybean meal, increasing from 17.5% to 29%, and in that way reducing the amount of corn in the diet. These were complex diets, so they did contain some whey and DDGS as well, in addition to poultry byproduct meal in this way. These components remained stable. All we're changing is by increasing the amount of soy, we're actually equalizing for SID lysine, methionine, tryptophan, threonine, and valine. So these 2 diets have equal concentrations of these first 5 limiting amino acids. The only difference here is the concentration of soybean meal. So that difference was made up using crystalline amino acids in that way. So certainly these diets are going to analyze different in terms of crude protein. We have a higher amount of soy, but what's important is that we've maintained equal concentrations of these amino acids, so trying to get rid of some of the confounding factors there. We had the good fortune to work with Dr. Mark Burhow at the USDA ARS over in Peoria, who has made his career in analyzing isoflavone content. So we sent these diets to them. They did this analysis, and so these are the analyzed concentrations of the first 3 isoflavones as well as their total. And what you can see is that we've increased in the low soybean diet from about 370 to 638 for genistein, the isoflavone which has been shown previously to have the greatest effects. So again, back to the Dr. Greiner's paper, between 200 and 400 milligrams per kilogram would be that sweet spot where we want to shoot. And even our low soy had that amount, and we nearly doubled that when going to the high soy. Now, just like with amino acids with minerals, we know that bioavailability can be an issue. And so even though we're showing the total concentration here, we know that what's actually available to the animal is going to be lower than that. So I present some evidence from a paper by Kuhn and others where they're showing that by increasing dietary isoflavones from soy from 7 to 450 milligrams per kilogram, there's a dose-dependent response on the concentration of those isoflavones in the blood. So we know that they are actually being absorbed by the animal and therefore should have a biological effect. So we looked at growth performance each week. Again, it was a 14-day study. Also looking at daily rectal temperatures. So all you graduate students, these are the fun things that you get, the fun measurements you get to collect. Starting at day 0 post-inoculation, so throughout that infective period. We also collected blood at days 0, 3, 7, and 14 post-inoculation for looking at immune parameters. So those included looking at total leukocytes, so complete blood cell count with the differential, as well as the circulating load of the PRRS virus as determined by qRT-PCR. We also then chose a limited number of immune parameters, including haptoglobin as an acute phase protein. So this individual protein is going to be increased very early in an infection, so it's giving us what's happening up front. In addition to that, we looked at a number of pro-inflammatory cytokines, TNF-alpha, interferon gamma, interleukin-1 beta, and interleukin-10. Interferon and interleukin-10 were not detectable in these samples, so we didn't see that response. What I'll focus on today are those tumor necrosis factor alpha and IL-1 beta as well was affected in this way. So from the stats perspective, again, we had 16 pigs, and that was those individual pigs were the experimental unit. The data were subjected to a 2A ANOVA using proc mix with repeated measures where appropriate. And again, we also looked at orthogonal contrast to differentiate within the infection status what was the effect of soybean meal there. So looking at that direct comparison. All right, jumping right into the results. What I'm showing on the left is looking at daily rectal temperatures. And this is including those which, in blue, were the sham inoculated pigs versus those which received the virus. What you can see is that the rectal temperature on each day is increased. And overall, we had a significant main effect here of PRRS infection. Basically, this increased their rectal temperature about 0.3 degrees centigrade. And this is very typical for what we see in this model. Shown on the right is looking at total leukocytes in the blood. And this is, again, is a very indicative response to this PRRS infection. No differences at 3 days post-inoculation. Those pigs which have been inoculated with PRRS, we see a dip and then almost an overcompensation as the body is actually activating that immune response. All right, so we know that we had a successful inoculation in this way. All pigs who were sham inoculated, we were unable to detect PRRS virus circulating there. And so here I'm only showing those pigs which were infected with PRRS. In the blue bar, we're looking at those pigs which received low soybean meal versus in the orange, those which received the high soybean meal. And here we're looking at days 3, 7, and 14 after we inoculated them. What's tricky here is the y-axis. So this is a CT value. This cycle threshold, this is by PCR, it's backwards. So the higher the CT, the lower the PRRS virus concentration. In this way, we see no differences due to soybean meal at days 3 or day 7. But when we get to day 14, we actually see a significant increase in the CT value here, suggesting that those pigs receiving the high soy had, had a lower concentration of PRRS virus. Excuse me. All right, haptoglobin, that acute phase protein. Excuse me, I'm fighting a cold. So we're looking here at 3 days post-inoculation. We've got the pigs fed low soy in blue and high soy in orange again. And now we're looking at both the sham and the PRRS, so all 4 groups of pigs. In this way, we saw a significant interaction between infection and dietary status. And that really stemmed from no difference here in the sham group, but a large difference here in PRRS-infected pigs. So the orange bar really doesn't change across these treatments. Around 1,300 micrograms per milliliter of this protein, so about a 4% increase only, but pigs receiving low soy, 121% increase. So that's at the early part of the infection. Now I want to switch to the later part. We're looking at over the entire period now, at day 14 post-inoculation, where we're still seeing effects of PRRS. We're seeing a very large increase here in the concentration of this pro-inflammatory cytokine. So no differences in our sham group, but we can detect TNF-alpha. This is always present. But in the induction with PRRS, we get a excuse me, much larger increase in those pigs receiving the low soy. So essentially, high soy has blunted that pro-inflammatory response, and we do see a significant interaction there again, with differences between low soy and high soy within this PRRS-infected group. Now, We also want to look at average daily gain. We showed that we've blunted some of that immunological response, and we see that no difference in our sham inoculated group. Again, those animals received diets which had equal concentrations of the first 5 limiting amino acids. But we see a larger reduction in growth in pigs fed low soy when infected with PRRS than high soy. So about a 50% reduction here from their control, and a 35% reduction here from 576 to 374. So in our view, the ability for soy then to reduce that inflammatory response has then translated into what we see in average daily gain over that entire period. Similar response in gain to feed in this way. So note that pigs infected with PRRS, they still eat, they still grow, but we're reducing that by about 50%, 40 to 50%. So in gain to feed, we're only seeing a reduction due to PRRS infection of about 18% here in feed efficiency. So to summarize, pigs fed the high soybean meal diets exhibited lower haptoglobin at 3 days post-inoculation, lower PRRS virus load overall, and lower concentrations of circulating TNF-alpha. This actually, it certainly reduced average daily gain in pigs infected with PRRS, but feeding the high soybean meal diet allowed those pigs to not lose as much weight. And overall average daily gain of pigs fed high soy tended to be greater in that PRRS infection group. So we think the implications of this type of research can be directly applicable to the swine industry. All we're doing is increasing the amount of soybean meal, be it in a weanling pig diet, where Dr. Stein just talked about some of the issues. We're here not in the first phase, but in the second phase. So if there's a farm that has a high incidence of PRRS, perhaps by increasing the amount of soy and realizing some of that added value, this may be potential. So inclusion of higher soy may reduce the severity of that PRRS infection. What we'd like to do is actually go back and look at both the duration of that infection and also the frequency. So there's lots of different ways to look at an infection in this way. So we believe that these nutritional strategies may have the potential to reduce performance losses due to PRRS and can therefore help to complement biosecurity protocols and other pharmaceutical interventions. We don't think this story is done yet. We haven't defined the mechanism for this, and I think it's important to note that though we had equal concentrations of the limiting amino acids, the non-essential amino acids are obviously going to be higher in that diet containing higher soy. So it's important that we go back and identify whether this is actually due directly to isoflavone content or perhaps to other factors, including that non-essential amino acid concentration. Additionally, I think that this has been done under very closely, under conditions in a very clean facility. So repeating this type of work in a commercial setting is also very important. Now, I don't have a picture of the graduate student who performed this work as Dr. Stein did, but Sam Rochel is the student who did this, and all other personnel. So if you ever get to see Sam, he's the one who got to collect those rectal temperatures on a daily basis. I also want to thank Dr. Jim Pettigrew for bringing me in on this project, and Dr. Dean Boyd for continuing this work. Analysis of isoflavone content by Dr. Berhow, and then the actual PRRS virus being provided by Dr. Van Alstyne from Purdue University. Finally, as with Dr. Stein, it was the Illinois Soy Association that funded this project, so we appreciate that funding from them. And apologize for my voice once again, but thank you very much for your attention.